conventional method here, going through the exploring what is the conventional understanding of corals and slugs at present, going on to aims, objectives, methodologies, results, and then putting that into a bit of context. So first and foremost, the conventional understanding of Scleractinians, oversimplification, but here we go. Most Scleractinians, or the majority, rely on zooxanthellae and the symbiosis with them to facilitate their energy gain. And though this is not all corals, this is certainly a majority, but all corals and all anthozoans rely on heterotrophy to make up the rest of this. So they actively have to feed on things. Now, one of the biggest conventional points is that the majority of corals rely on the heterotrophy of particulate organic carbon and things like microscopic plankton, phytoplankton, and zooplankton. Switching organisms entirely, sea slugs are things that are considered toxic and rather pretty. In this case, I'm talking about sea slugs in what used to be considered the opisthobranch taxa. This is no longer a recognized term taxonomically, but they do have shared ecological and biological characters, among which is toxicity. So they feed on a very specific range of prey items that give them the toxicity that they use as chemical deterrents for their defenses, to the point where there are no known obligate predators of sea slugs today. The main predators of sea slugs are the nudibranchs and other sea slugs. There have been some cases of sea spiders, sea turtles, and cone snails that have opportunistically fed on them, but nothing fixed. So let's challenge that a little bit. First and foremost, there are records of a few coral species and families predating on certain larger planktonic prey, so jellyfish and salps. This is in the Dendrophilidae, Euphilidae, and Fungidae. And then moving on from that, in 2015, we observed a coral eating a slug. We observed an ingestion that lasted about 20 minutes, well, ingestion, and then waited to see what would happen next, and then we ran out of air in our tanks. So we didn't see what happened next, right? But we do know this was an in situ observation. We had nothing to do with this. There were no humans involved in the water prior to our arrival. So this was observed at Koh Tao in the Gulf of Thailand. This is where I do most of my work. And at the same location, following on from that, we observed a different coral, Heteropsamia cochlea, ingesting another kind of sea slug called Tubulophilenopsis pillsbury, and actively predating and catching prey on Costacella sea slugs on the macroalgae Avonvillea. So Heteropsamia cochlea thrives and is exclusive to the soft sediment habitats beyond the coral reef. They are not found within the typical fringing coral reef, whereas the fungids that we have observed are within the lower reef slope and the reef base or reef edge. So we find one fungid species out in the soft sediments, the majority of which make up the reef building coral on the fringing reef. So putting all of this together, we have 4 very specific questions that I'm going to actually read out. Are sea slugs actively consumed or just opportunistically ingested, i.e. maybe they're rejected? Does slug size play a role in ingestion, rejection, or consumption by a predator organism, predator coral in this case? Are there differences in predation between different corals, such as different species or different feeding morphologies? Does habitats such as reef or soft sediment habitats influence capacity of slugs as prey for associated corals of these habitats, i.e. Heteropsamia being representative of the soft sediments? So essentially, we fed slugs to corals. We tried 6 different species of Sacoglossan sea slug. So the Sacoglossans is just like the initial observation in 2015, which was a Placobranchus sea slug in the mouth of a Ploractis pomatensis fungid. So we tried 6 different Sacoglossans. 3 of those are specific to typically the fringing coral reef, and 3 on the soft sediment habitats. And then we tried, we had a broad spectrum of sizes, so we took individuals of all sizes based on what we could find in situ, and we fed them to 4 different corals. So we fed them to 3 free-living mushroom corals, which is Ploractis pomatensis again, like the initial observation, Fungia fungioides and Dana fungia scroposa, and we used Heteropsamia cochlea as the 4th, which is the dendrophilid. We measured the mouth diameter and the max diameter of the coral to see if there's a correlation between these sizes and the predatory capacity of these corals. This is just the beginning of a long list of measurements. So here are the 6 slugs. The first 2 are Costacella, and Elysia japonica over there on C. So these are the three that are exclusive to the soft sediment habitats. We do not see them in the coral reef. We have Elysia pusilla and two of the Placobranca species that we tend to find in the shallow reef environments. So 40 individuals collected in situ were fed to each of the different corals. So we have 10 for in situ mushroom corals, 10 for ex situ, 10 for in situ Heteropsamia, and 10 ex situ. All trials were conducted during the daytime. We tried to conduct them at night, led to some interesting problems. You can ask me about that later. And all the in situ trials that were carried out at the representative depths of these organisms. So 4 to 6 meters for the fungids and 11 to 13 meters for Heteropsamia. And the ex situ trials were carried out in aquarium tanks in our little office. So here are some visuals of what it looks like when a coral eats a slug. That's Elysia japonica over there being predated on by Heteropsamia, and Elysia pusilla being predated upon by a fungid. So, measurements that were taken. How long it takes for the coral to ingest the slug. So once the slug has been placed upon the mouth, how long does that ingestion actually take? How long until rejection, if there is rejection? We measured the slugs prior to ingestion at their max length and divided all the slugs into 3 different size classes, so less than 6 millimeters, those between 6 and 12 millimeters, and those larger than 12 millimeters. We had a post-ingestion observation time in situ of about an hour, basically dependent on how much air we had in our tanks. Most importantly here was we assessed the consumption by tissue degradation post-ingestion. So tissue degradation was assessed upon rejection, if that happened, and in pretty much all cases, rejected slugs were largely undigested and crawled away slightly confused. And the post-ingestion, fixed intervals post-ingestion were when the slug was extracted from the coral itself, and this was assessed. So it was given us 1 of 3 scores, 0 if the extracted animal was largely undigested or unharmed, 0.5 if there was partial digestion, and 1 if there was heavy digestion. This was arguably subjective, but this is what it looks like. So, relatively undigested, not had a good day, heavily digested. So we used Crystal Wallace and Wilcoxon's test for significance and Pearson's to check for correlation. So, what we found. First and foremost, 3 of the species that we tried did show regular and reliable instances of consumption. In fact, Elysia pusilla over here was partially or heavily underwent partial or heavy tissue breakdown after an hour in almost all instances, with the 2 Costaciellas showing some degree of tissue degradation. So these are mean score values. This is an average of that 0 to 1 scale that I provided earlier. And all 3 of these species are just, whether it's a coincidence or not, these are ones associated with a very specific host macroalgae that they are found, where the other 3 species are free living. What that implication is, we're not sure yet. We need to do a lot more work. The other 3 species, minimal to no tissue degradation, so they are actively ingested, but then spat back out. So the mean consumption with regarding to prey size, the smaller the slug individual, the more heavy the degradation, and if you get larger than 12 millimeters, the coral's basically not having it. Similar trend with in how long it takes to ingest a slug, the ingestion time. Smaller individuals are ingested faster, larger individuals take longer to swallow. So, very quick summary here. As mentioned, we found significance in consumption, ingestion, and rejection with regards to prey species. With regards to sizes, consumption and injection, but regardless of prey size, the slugs were spat out at the same speed, which is no significant difference in the rejection. Between the Heteropsamia, representative of the soft sediments, and the mushroom corals, we found there was no significant difference in the consumption, so the palatability remained the same. However, there was a difference in how long it took to ingest the animal and how long it took to reject a given animal. There was no correlation found between the size of the mouth and the size of the overall coral, because once again, these are free-living single polyp corals. With regards to their palatability, with regards to their predation of the slugs. Why we found some significance in the rejection between the different fungid species, we do not know. We will explore that. And we did find a little bit of difference between in and ex situ trials. This is, even though we did our best to mimic the natural environment, there was only so much wave action we could do with our hands, etc. And wave action is particularly important for things like mushroom coral. But we will look into that further too. So, what does this, it's fairly simple conclusions. Sea slugs, at least at one location, are viable prey items for Scleractinian corals. Though we know that Scleractinian corals are opportunistic predators, they are not limited to planktonic matter. This is based now on both natural in situ observations and their ability to consume certain sea slugs. These are all benthic associated animals. None of them are demersal. Some of the most widespread and significant predators of sea slugs may be hiding in plain sight, the reef-building corals of a coral reef. So what I would like to end on is this. We tried several species of slugs with several species of corals outside the basic data collection that I've shown you. So we have corals such as the flabellid corals, Truncata flabellum. We tried Heterocyathus. We tried a few other sarcoglossans based on availability and seasonal availability. And we found a wide variety of trends. So none of this is conclusive. The data was not really taken on many of these, but there is definitely more to be explored here. We tried a couple of nudibranchs. They appear to be quite tasty for some corals. So on that note, I will shut up.
Thank you very much. So questions from the audience, yes, please.
Very interesting research. And my question is, in the real world, what do you think? How come the sea slugs jump into the fungid corals? Do you also have an observation about that? Or probably the coral have a attractive signal for them? I don't know.
Well, I certainly don't think it involves jumping. I will go that far. Well, I'll take us quite far back, if I could, way back at the beginning, where at least for the Costacielas, the two green ones that, oopsie, this is on a mind of its own. Just over here, we can actually see, you can't actually see it very clearly, but within those red circles are individual sea slugs. So the tentacles of the Heteropsamia are well within reach of many of these animals, and the moment that the tentacle comes in contact, it will grab onto it. How the fungids do it, I'm not entirely sure. Once again, doubt it has anything to do with jumping. However, I will say that many of the in situ trials that we did have, the moment that the slug was caught by the coral, moonwrasse, amongst others, would come in and carry out kleptoparasitism. They would try and ingest the slug only once it had been caught onto the coral, take it into their mouth, swim away and realize they didn't like it, and then spit it back out. And more often than not, that would fall onto another coral, typically a mushroom coral. So it may just be that opportunistic predators such as fish will pick them up and move them around, and it happens to land on mushroom corals. Kotal has a very high abundance of mushroom corals. Beyond that, I cannot say that is something we definitely need to look into, but once again, these are all corals that can move around. There is mobility involved here. So, yeah.
They hunt.
I don't know if they hunt, but they definitely are hungry.
Thank you very much. So that would be enough for the presentation of